Hi everyone, thanks very much for joining in. Welcome to our webinar today on Conservation International's recently published Core Standardized Methods for Rapid Biological Field Assessment. The webinar will outline the contents of the handbook, which provides reliable, standardized and replicable methodologies for quickly assessing key ecosystem values in the field. The handbook was developed with input from over 60 experts and was supported by Chevron. It provides useful technical guidance for conservation planning and decision-making at the local to regional scale. Following this, we will finish with a Q&A. So my name is Anna Gray, the Environment Coordinator for IPECA. I'm just going to briefly introduce our speakers today and then run through some technical logistics. Today, our webinar will be presented by Leonardo Viana, Trond Larsen, and with an introduction by Sarah Connick. Leonardo Viana is the Director in Conservation International's Responsible Mining and Energy Program. His role is as Principal Scientific Advisor to mining and energy companies in support of CI's field and headquarters-driven global partnerships. He contributes expertise on issues including biological assessment and monitoring, biodiversity offsets, mitigation hierarchy, and achievement of net positive impact, and international performance standards. Leo has a diverse background in the consulting and non-profit sectors. Dr. Trond Larsen is a conservation ecologist who has studied tropical biodiversity for the past 20 years. As Director of Conservation International's Rapid Assessment Program, Trond leads biodiversity assessments to guide conservation priorities and decision-making at sites around the world. He is an expert in assessing the impacts of anthropogenic change on biodiversity and developing appropriate conservation recommendations. He has experience in a range of sectors, including extractive activities. Trond also holds a PhD in Ecology and Evolutionary Biology, is a research collaborator at the Smithsonian Institution, was a founding director of the Amazon Conservation Association, and worked as Science Director for Osa Conservation in Costa Rica. Sarah Connick leads the Environmental Stewardship Team within Chevron's Corporate Health, Environment and Safety Department. She is responsible for managing, providing business support on Chevron's internal environmental processes, standards and tools, as well as external engagement on emerging issues related to biodiversity and natural resources management. Prior to joining Chevron, Sarah worked on a range of environmental issues in the governmental and non-profit sectors. She holds a bachelor's degree in chemistry, a master's in environmental engineering, and a PhD in environmental science, policy and management. And of course, Sarah is also the chair of IPECA's Biodiversity and Ecosystem Services Working Group. So before I hand over to Leo, Trond and Sarah, I'll quickly run through some technical logistics. Everyone is currently on mute. During the Q&A session, you can either raise your hand via the WebEx system to indicate that you would like to ask a question or make a comment, after which I'll unmute you, or you can type a question into the dialog box. You can either send a question to all participants, just to myself or the other presenters. I'll then read these out during the Q&A. So now that's all been said, I can hand over to our speakers for the presentation. Sarah, over to you when you're ready.
Thank you so much, Ruth. So I just wanted to provide a little bit of background on the Chevron-CI partnership and the origins of this document that Trond and Leo are going to present. Chevron has worked with Conservation International now for, I think, over 15 years, beginning with the Energy and Biodiversity Initiative in 2001. And more recently, we established a 5-year strategic partnership and are currently in year 3 of that. And as a part of that partnership, we identified 2 objectives, and I'm going to talk about the first one here, which is improving identification of environmental risks and opportunities. And one of the things that we had been looking at, in Chevron, I think that folks are probably familiar with this from the Cross-Sector Biodiversity Initiative as well, but the need for really good quality biodiversity baselines that can deliver consistency and replicability. And what was great was that this matched up really well with CI's interest in taking its approach to rapid biodiversity assessment and really codifying what the key methodologies are for doing that. And in this case, these are field methods. And so this, I think that this document serves as a real complement to the CSBI document on developing biodiversity baselines. To date, Chevron has not yet had the opportunity to use these methods on a major capital project, but we did use them recently. Conservation International applied them in a community-based watershed restoration project that is underway in partnership with our business in China. So with that background, and I can answer questions at the end as well, I'd like to pass it on to Trond and Leo. Thank you. You may be muted.
I'm sorry, can you hear me now?
Yes.
Great. Thank you, Anna and Sarah, for the introduction. I wanted to, before we get to the book, just set the stage a little bit about the importance of having such a publication. Since the Renaissance, people have said what gets measured gets controlled, gets managed, and ultimately gets improved. If you can measure it, you can do something about it. A little bit about my background. I was a conservation biologist and was doing environmental impact assessments, and commonly came across the difficulties working in mining and energy projects, and came across the difficulty of, if you were working with one consultancy firm, you would do something in one way, using one methodology. If you were working with a university professor, he may have his own techniques and would do it some other way. So quickly you can see the difficulty in trying to compare results, gauge your processes, and gauge the things that you're doing. So just to leave with that thought of, what gets measured gets controlled, and gets improved, and gets ultimately improved. In addition, we currently have these different drivers. We find ourselves in a situation, what gave birth to the idea of a book like this. We find these different drivers that are driving us to improve our techniques and our approaches. Within businesses, we have corporate policies now that are mandating that business units have a method in place, a standardized method in place for collecting, in this case, baseline assessment data. But also governments, regulatory drivers that are all too prevalent are requesting that as well. There are untold stories of processes that are done, procedures that are done, best efforts sort of hindered because as a corporate member, you would hire a consultant to do something in a certain way, and then you would submit that work, and then regulatory agencies would say, well, it wasn't done to this standard, or this wasn't done. So again, these are the impetuses for us in sort of putting together this publication. And lastly, as many of you may know on the phone, there are drivers that come from financial lenders, such as the IFC Performance Standard 6, Equator Principal Banks. So all of these are converging to sort of require that we think more holistically and more standardized about some of the methodologies that we utilize. Now, don't be frightened by all of the text on the screen. What I want to draw your attention to are the different lines. So in the previous slide, we had drivers in orange, government in green, and lenders in blue. And here you'll have the top line there is a typical project timeline where you have where you go from identifying and assessing exploration all the way to closure and decommissioning of said project. And so forth and so on. There's the mitigation line, which is in green, which is the regulatory line, the lender line, which is in blue, and underneath all of those lines in gray is the mitigation hierarchy, which many of you would be familiar with. I'll come back to this slide in more detail, but what I want to point out are the little yellow circles there that are moments along all of those timelines in which collection of data is very important. So just to keep that, to show you the sort of overlapping drivers and the need for data. Thus, looking at the drivers, looking at sort of project timeline needs, you quickly become aware that assessing biodiversity components of an area for decision making is of paramount importance. But current scenarios exist. As I mentioned, I touched upon them a little bit. We lack an international consensus for most taxonomic groups, so there's that sort of starting difficulty. And we also lack detailed technical guidance. Now, I say technical guidance, and I mention that because Sarah just brought up a great point. The CSBI guidance, and like most guidance, like the IFC Performance Standard 6, those are practical. Those are guidance in what you should do, but not how you should do it. So I think this publication stands apart in that it specifically tells you how to do what you need to do, how you go about collecting the data for these 12 taxonomic groups that we've selected, and Trond will talk about a little later. So it's meant to be sort of a complementary piece to the other guidances that are out there, like the IFC, like the CSBI. Because most methodologies to collect baseline data are not standardized, as I mentioned, with researcher bias, and I felt that firsthand in the work that I did in the Amazon. And also that work, the methods that are currently used many times are just about presence or absence. Researchers will tell you, yes, you have this species on your site, but what you really need to know to sort of meet regulatory requirements is something about the abundance and how are the species doing, not whether they're just there or not. And these types of standardized data will give you that richer, fuller abundance and population data. Also, another barrier with current methodologies is that they're unable to be replicated because you're doing these preferred methods by one researcher that may not transition to another area of the world where you have, if you're a large enough corporate entity, you'll have sites all over the world, and that nuance between capturing a certain type of species in one place versus another place isn't captured in these regional researcher methodologies. So therefore, all of these barriers lead to sort of a lack of robustness of your data. You've put in the effort, you put in the time, but you cannot sort of provide the information that regulators are requiring, or you yourself may be requiring in terms of being able to compare the performance of a certain site. So with that, I'll turn it over to Trond to talk about the actual publication.
Thank you, Leo. Can people hear me?
Yes.
Okay, great. Yes, I'll talk a little bit more detail about the handbook itself. We really think that this is the first book of its kind for several of the reasons that Leo mentioned already. There are a few books out there that provide detailed methodologies for collecting data for a wide range of taxonomic groups. But all of those books and all of that guidance are essentially review materials that cover, the huge range of methods that exist, which is great, but it's not practical in the sense that it tells you, okay, here are the specific methods you should choose, because obviously you can't go and employ every approach out in the field when you're conducting a baseline survey or doing some other type of rapid survey. So the goal of this was to have essentially a one-stop shop for multiple different taxonomic groups that really focused on those essential core methods. So we looked at the wide range of all the methods available and asked a variety of researchers to come to a consensus on what those core methods that should be used everywhere and employed by everyone should be. Those methods had to be standardized for the same reasons that Leo has already mentioned. And they also need to be applicable globally. Now, I will mention that this handbook is primarily focused on tropical systems globally, but its applicability is really broader than that. The tropics are perhaps the most challenging place with their high diversity and highly varied environments. So just briefly, people are probably familiar with what we mean by standardized methods, but I'll just give a few examples to clarify that if that helps. they provide very clear, pragmatic sampling parameters. Some examples of these are, so you essentially want to delineate where across space you're measuring whatever group you're focusing on in a way that can be replicated later and by other people and in other places. So for vegetation, for example, you measure out a square 1 hectare plot, and you can measure plants and trees of all certain diameter range that you choose within those plots. You can have a gridded array of camera traps that are all equally spaced. You can conduct line transects, which are essentially walking transects, where you record all birds or primates or other species within a certain distance of the transect that you're walking along. For insects and amphibians and some other groups, equally spaced transects of pitfall traps. Same type of approach for freshwater and fish. So essentially, I think you get the idea that it's really trying to approach something that can be replicated and understood easily. I'll just let Leo talk quickly about this slide before I jump back into the next one.
Thanks, Tron. So coming back to the slide I showed you earlier, I just wanted to point out let me just ask, can everyone hear me?
Yes.
Great. Okay, just wanted to make sure. So along the project timeline, you have those, for many of you on the phone will be familiar with the project timeline steps. And then you see here in these sort of yellow, the little yellow circles, where we've pointed out from at least from our point of view, and you may have more instances, where the data collection is really important. So when you're executing and you're doing construction, having a sense of the baseline for your area is very important. And that lines up really well with the regulatory mitigation within sort of an ESHA process or an EIA process, where you're monitoring for environmental impacts. Oh, sorry, I should step back and say, when you're executing your ESHA here in the green, that first circle in the green there that you have, let me see if I have a pointer. Does that work?
I don't think there is a pointer, Leo, but I think, oh, he has got a pointer.
Is that a pointer? This is
yeah.
webinar 2.0. So when you're looking at this first moment, you have to set up your baseline, and then when you move to executing, you're also monitoring environmental impacts, those that you've identified in your baseline that you're going to track. So you're going to track that the length of the project cycle through your operations and through closure and decommissioning. Meanwhile, if you've utilized any lending or borrowing, you're also going to have compliance monitoring for the life of the loan too. So I just wanted to sort of be a little bit more in depth in terms of all of the different instances, because we talk about this being for biological field assessment, but there's also ways that you can utilize these methodologies to keep track and sort of monitor some of the species that you've identified as important to your particular site. Let me just take that off, and back to you, Tron.
Thank you, Leo. This I think might be the most important slide to focus on for this presentation. No pretty pictures to look at while I talk, unfortunately, but I will spend a little bit of time explaining each of the points in this list of what we see as the major benefits and advantages of having this type of practical handbook available. So I'll start out just by saying that I am a field biologist. I collectively have spent many years of my life in a tent in the forest somewhere around the world, and that's what I really love to do. I've worked with a lot of different really incredible and very experienced biologists whose knowledge is unsurpassed through many decades of experience. And sometimes you talk to these people and you ask them about standardized methods and standardized approaches, and some people do immediately sort of cringe when you try to mention that type of approach, because they are so experienced and knowledgeable that they're able to, for example, enter a forest, walk around. They know exactly where they're likely to find some of the hard to find or rare species, whether it's by sound, whether it's by looking in certain microhabitats. And they can maximize the species list of things that they find in a given place, especially where no data have been collected before, simply by tapping into that experience and knowledge that they have. And really what you would call sort of a shotgun approach, where you just go wherever you find appropriate places and look for what's there. And I just really want to emphasize that there's no replacement for that type of experience and knowledge. And this handbook is not intended to replace that. What it's intended to do is provide a complement to those approaches that are standardized and is a core methodology. So I just want to make that very clear, that we're not trying to replace all of that knowledge. So, okay. The first benefit I'll talk about is being able to monitor changes in biodiversity over time, and Leo just alluded to that in the previous figure. Again, if you're not using some core standardized approach, it becomes very difficult to replicate the same methods so that you can truly understand if species are improving or populations are declining over time. Again, without standardized methods, it's very difficult to compare data from different sites. So when you're using quantitative and similar approaches, it allows you to much more easily place the data from a baseline assessment at a given site, for example, within a regional and global context. So you can compare simple species lists, and that's useful, but it doesn't necessarily tell you, let's say a harpy eagle flew over randomly at one site while someone was measuring it, and that goes on the list, whereas another site has some major nesting populations of harpy eagles, that you wouldn't necessarily see those differences. So that's one reason that this approach can place those data within a context, a broader context, which is really important. And again, this has been mentioned, but the ability to collect population-level abundance data is really critical. A lot of the changes and impacts on biodiversity are not simply whether a species is present or not present in a place. They don't tend to just completely disappear. They can in some cases, but it tends to be changes in the overall population. And so these kind of standardized quantified approaches can yield those types of data that are really essential for detecting change and understanding trends. Standardized approaches also make it easier for a wider range of users to adopt those methods, rather than just needing to rely on those handful of global experts that are the leaders in their field, but aren't necessarily always available. They can't necessarily cover all of the sites where things need to be measured. So this approach makes it easier for other people to replicate similar approaches. Another really key point is that we had to walk a line in coming up with these approaches that identified standard methods that could be applied globally, while also understanding that there are certain regional and geographic contexts that will affect how you need to sample your taxonomic groups. And so that is built into this handbook, is that there's not one single approach. It's a combination of a single approach, but with modifications depending on where you are in the world and what type of biome or habitat you're in. So the next two, cost effectiveness and sampling completeness, are related. So when I say sampling completeness, I want you to look at the figure on the left here, which is a species accumulation curve. So on the vertical y-axis is the number of species that you found at a given site, and the horizontal x-axis is your sampling effort. In this case, it's measured as the number of individuals collected or observed, but it can be measured in days in the field or in other number of traps or other types of ways. But essentially, what it can tell you is how exhaustive your sampling has been. So if you spend a week at a site, which is often how long people do spend in rapid surveys, have you sampled 100% of the species there? Have you sampled 5% of the species there? It can vary greatly. So without being able to construct these types of curves where you can see when those curves begin to reach a leveling plateau, you really don't know whether you've described accurately the biological community there. And so using these standardized approaches allows you to construct these curves and actually estimate what % of the total species at a given place you've found. And finally, as much as possible, we wanted to include both innovative and automated approaches. Automated approaches are really good for eliminating researcher bias. Things like camera trapping, bioacoustic recording, where you can just put out a box that records calls 24 hours a day. You can do it for all sorts of things, birds, bats, amphibians, katydids, other types of insects. And I won't really talk about metagenomics because it's not in this book, but it is an approach that we're exploring and have started to use, where, for example, you can collect leeches and grind up the DNA from the blood that they fed on and identify all of the different vertebrate species in an area that you might otherwise never find. Okay, so one of the ways we wanted to kickstart this was to hold a workshop where we could bring together as many of the experts from different parts of the world into one place where they could sit down in their working groups and really come to a consensus, because that's one of the most challenging parts of this exercise. Everyone has different approaches. Biologists can be very stubborn about using the approach that they like and have used in the past. So this was, I think this workshop worked out very well. We had some 40 people or so, I think, present. And there was, some lively discussion, but by the end of the workshop, people did reach a consensus on what those core standardized methods should be. And then, of course, there was a lot of preparation before and after, and a lot of the writing went in before and after the workshop. But this was a way to bring people together to identify that consensus. So here you can see the table of contents of the groups that are covered in the book. At the moment, it's 12 taxonomic groups. We covered basically most of the plants and the vertebrates, which are the major groups. And then you can't measure all invertebrate taxa. There's simply too many. So we tried to pick a subset of those which can serve as indicator groups or that are particularly ecologically important in an ecosystem, or might be represented by threatened species. So those are why some of the invertebrates like butterflies and ants and mollusks and dung beetles are on the list. So there was technical guidance from 65 experts. There were 34 authors and 31 peer reviewers. So it was a very extensive peer review process, and we really thought this was essential, again, for building that consensus and not just representing the views of a few people. And this also helps to ensure wider buy-in by the broader community in employing these types of methods. So apologies for the small text here. I don't expect you to necessarily read this. I'll just briefly go over the structure. So here is the guidance that we gave to authors about how to approach writing their chapters. So you start with the introduction and what is the taxon of focus, and most importantly, what are the criteria that make that taxon so important in rapid baseline surveys? Is it an indicator of water quality? Is it an indicator of the health and biomass of the large mammal community? Is it cost effective to sample? Those kinds of criteria. Then the bulk of the chapter is simply a description of what those core standardized methods should be. Practical guidance like the number of people that you need, how long they need to be there, what types of approaches you need to employ to measure abundance and population size and density. We have box lists of exactly what types of supplies and equipment would be needed in the field, both before and after the field work, including follow-up work in the lab. How do you go about selecting the sites where you're going to do your sampling once you're in the field, trying to maximize the variety of habitats that are present, the sampling effort that would be required, so again, through these types of species accumulation curves. Sorry, and context-dependent sampling considerations. I already mentioned this, but different types of environments, whether it is a wet forest or a very dry forest or scrubland, is going to change. For some taxonomic groups, it's going to change how you have to set up your methodology and your trapping, for example. So we wanted to build these options into each chapter and guidance. Biogeographic differences, I'll give you an example. In the New World, in the Americas, the primary method to sample bats is using mist nets, but in the Old World, where they've evolved from a different lineage and they have different types of echolocation, they don't get caught very often at all in mist nets, so you have to use an entirely different kind of trap called a harp trap. And that's just a biogeographic difference, but it's very relevant and important to understand in the guidance. Seasonal considerations, some groups are much more affected, for example, by dry and wet season considerations than others. Things like butterflies are very dynamic and seasonal over the course of the year, so that needs to be understood and placed within the context of your sampling design. In addition to the core methods, we wanted people just to quickly assess what additional supplemental methods should be used. And then there's some guidance on data management, how you deal with specimens identification and processing, et cetera, and then concluding with what the implications of the findings are and how they can be used, and also what the constraints and limitations are for rapid survey. Because again, rapid surveys, you're not going to be able to measure everything. So what are the trade-offs and what can be gained? That's the bulk of it. Just at the end here, I'll just run through a few slides to give you an idea of what's included in some of the chapters. So for vascular plants here, just to show you some of the diagrams from the chapters in the book, so you can see where you're supposed to measure on a tree, above the buttresses of the tree, if there's deformities, if you're working on a slope. There's all different approaches depending on the types of trees you're measuring. And this is relevant also for assessing stocks of carbon using on-the-ground field measurements. And then on the right, you can see the blue lines are the transects that you would walk to locate and identify plants and document the diversity of plants, and how those would need to be modified depending on the terrain, barriers like rivers and mountains, those kind of things. And again, not to read through any of this text, but for vascular epiphytes that need different approaches than other types of plants. Epiphytes are those plants that are growing higher up onto trees, hanging onto trees. So, a practical box demonstrating what your target habitats would be, what types of data you gain, the time and personnel needed, the skills required, the equipment, and then guidance about where to sample at each level within the forest, and how to do it in each level. So again, don't need to read that, but just to provide some examples. For medium and large-sized mammals, You can see on the left an example of a line transect, which is a very effective and standardized way to measure the presence and abundance of large, medium, and large-sized mammals. So you walk along this transect, you can see the green lines there, and you record everything that you see or hear within a perpendicular distance of 90 meters. Camera trapping is fantastic, again, because it's automated. You can leave the camera traps out for a long time without needing to do anything except for collect and maintain them if the batteries need replacing, and then finally analyzing your data at the end of the study period. You can see the three images there are an example of the sampling design process. You start with a map of your study location, your different habitat types. You can see rivers there. And then you put your optimal grid of where camera traps would be if they're all equally spaced across the entire study site, which are all the points on the map there. And then you have to refine that based on practicality of where you can actually walk, how the terrain allows you to access those places, so you have a smaller set of locations where you can put those camera traps in the end. And here's also an example of species accumulation curves for camera trapping based on how many cameras you've employed. So this can give you valuable information. Do you need to buy 10 cameras for your survey, or do you need 60 cameras? In most cases, you do need more, and it also tells you how long those need to be deployed in the field. For example, typically camera trapping needs to be done for 3 or 4 weeks minimum, whereas many other approaches can be completed in a week or less at a site. Bats, as I mentioned, there's just a photo of what a HARP trap looks like. In the New World, we use mostly mist nets. Again, not to read here, but for small non-volant mammals, primarily rodents, possums, things like that, there's examples of exactly what equipment and supplies one should buy for any place that they're going to be sampling in the field around the world. So I won't go through that list. And then there's, a variety of things that you need to actually prepare specimens for museum collections and for further identification. The bird chapter includes an overview of the most common approaches to surveying birds, what their strengths and caveats are, and then focusing in on what that core standardized set of approaches should be, which are point counts, line transects, and mist netting. Herpetofauna, so reptiles and amphibians, just a few images of some of the standardized measures. You can put in pitfall traps. You can see on the left along linear lines with fences that sort of funnel the reptiles and amphibians towards those pitfall traps. And then that green box with a frog sitting on it is a bioacoustic recording device that you can just leave out for months at a time, and then later be able to analyze those recordings and identify which frogs are there, how frequently they're calling, and those types of data. Fruit-feeding butterflies, just a diagram of a standardized trap design. Fruit-feeding butterflies are a major component of the overall butterfly community, and they can be sampled using bananas as bait, fermented bananas, so you can do that anywhere in the world and quantify the butterfly community and compare those data very easily across sites and over time. Finally, freshwater mollusks, again, an indicator of water quality. There's several different approaches. There's qualitative sampling, as you can see in a photo here. There's much more rigorous approaches like quadrats that you drop to the bottom of the water body and just measure all of the mollusks within that square, collecting tissue samples for genetic analysis, etc. So concluding, the handbook has guidance from, based on input from actually more than 65 experts when you include some of the reviewers that weren't necessarily the scientific peer review community, but that also put a lot of work into this. Provides detailed methodologies for 12 taxonomic groups. We do have ambitions in the future to expand this to marine environments, and also perhaps to consider a similar approach for measuring ecosystem services. It can be used at different stages, impact assessment and other stages of the ESHIP process. Describes how much sampling is really needed for each taxonomic group. Provides very practical advice, like equipment lists, what the limitations are for the surveys. And while standardized globally, these methods are also tailored to specific places where regional differences require it. And finally, it's useful for implementing sampling efforts or assessing the quality of baseline data collected. And so we can even do this sort of retroactively, looking at studies that have been done in the past if we're to repeat studies now. So that is all from me. You can download the handbook from this site you can see here. You can get in touch with us. Also, if you're interested in a copy, it's also available for sale from the Natural History bookstore in the UK. Thank you, and I think we're all free to take any questions now.
Yes, Trond, Leo, thank you so much. That was a really interesting talk and lots of really great pictures as well. So we have a few questions come in, so I'll dive right in, but if anyone else has any more, please do send them over on the Q&A box. So firstly, Trond, you just mentioned at the end there about how you're thinking of developing maybe a similar kind of thing for ecosystem services in the marine environment. What do you think would be similar for this, and what would the big differences be, and do you think it would be a similar kind of format?
For a marine, I think it would be pretty much the same format. We've already started this work for marine taxa. We've had many scientists interested and are on board, but we haven't gotten to the stage of putting together a workshop and writing chapters for it yet. But essentially, the structure and the approach would be very much the same. For ecosystem services, again, the overall framework would be similar, but they're very different in how you measure it. There's much more household survey type approaches involved and other types of work.
Great, thanks. And I have another one asking about what's the uptake been by companies versus consultants with the handbook? Do you know?
We are still rolling this out with various webinars and presentations and attending conferences. So this is all still relatively new. So I don't know if Leo or Sarah would like to respond to that also.
Yeah, I can jump in here, Trond. This is Leo. We've, as Trond mentioned, we've rolled this out in a couple of different places. The SPE conference in Norway was our soft launch in April. So we've had other touch points, other webinars that we've given both internally to corporations, but we've also sent out a newsletter to a select number of folks, and the analytics on that newsletter, where you can see who clicked and who opened and who downloaded, has it pretty much evenly distributed between corporations and consultants. To touch upon that point more broadly, we see it as a tool for both. We see it as a useful tool for the environmental manager at a site that that is tasked with having to do a baseline assessment, or tasked with, as Trond mentioned, maybe revisiting a baseline assessment to see if what was done correctly. This is a tool in their toolbox to be able to assess the quality of a consultant that they may hire. And then the vision would be that consultants everywhere would welcome a standardized approach that they could evaluate all the processes that they do. I can say from personal experience, I used to work at ERM, Environmental Resources Management, and the volume of work sometimes is much. So having standardized approaches for a subset of work that you do really facilitates and makes it more efficient.
Great, thanks, Leo and Trond. It's great to hear more about consistency between approaches. I have another question here. Do you recommend any periodicity of monitoring for all the taxa you mentioned?
I'm not sure if this is referring specifically to seasonality and sampling over the course of a year, or whether it's asking about whether you should monitor at a certain number of year intervals.
I think probably seasonality.
Seasonality. So yes, we asked everyone to address this in every one of their chapters, and for some taxonomic groups, it's much more relevant and important than for others. Mammal activity, for example, might increase or decrease depending on the wet or dry season. Certainly in temperate areas, obviously, you're not going to be going out and sampling in the winter. But we do highlight the areas where it's most essential, and that's, for example, in freshwater environments where water levels can change drastically, and your ability to catch fish is hugely affected by seasonality. So in those cases, yes, we strongly recommend, if possible, to do both wet and dry season sampling. If you can only do it at one time point, then you do it during the period when you're likely to be able to sample the highest number of species.
Makes sense, thank you. I have another question. Is it still possible to compare standardized methods effectively when they've been tailored to specific geographic needs?
Yes. Again, this was a really hard balance to strike. It was necessary in many cases, but I would say it's a handful of examples where those methods really need to be tailored for different environments. If you're talking about a forest versus a savanna, that for example can affect how you can sample. But as long as you're using the same approaches in those different habitat types, it's still very comparable. In terms of biogeographic differences, again, you can still compare things like species richness and abundance between harp traps and mist nets. But it just doesn't make sense to use a mist net in a place where you're not going to catch any bats, or to use a harp trap in a place where you're not going to catch any bats. So yes, it's comparable, and there's a balance.
Great, thank you. We have a few minutes left. I'm going to call on Sarah to maybe give us some examples or give some more reflection on how it's been using this handbook in Chevron, if you've got any insights for us.
Yeah, so as I mentioned earlier, we have not yet applied this directly on a Chevron project in the field. The one experience we have with it is we have a partnership on a watershed restoration project in China, for which, in doing the initial status assessment of the watershed, Conservation International used some of the methodologies in the handbook. And so we're hopeful that, once progress has been made on restoration efforts, that we can go back and replicate those and see some positive change.
Great, thanks, Sarah. Trond and Leo, have you got any examples where you know that some companies have already started implementing the techniques you've described here?
This is Leo. No, currently, as I said, because the rollout was in April, we've not, with the exception to the case that Sarah mentioned, we're in a phase of sort of amplification where we're sort of getting people to know about it, getting people to understand it and discussing it. But as Tron mentioned, and as we said here before, it's based on methodology that the rapid assessment team here at CI has utilized, many years. So we don't believe that, the methodologies will be questioned. It's just a question of getting uptake. I think it's getting people to see the value in it.
And I also can just chime in and say we're seeing a lot of interest from people in learning more about this. The distributor that we've been using in the UK has sold quite a lot of these books already. So we don't know necessarily whether they're being used already in the field, but we know that a lot of people have been accessing these methods and reading about them.
Great, thanks guys. And yeah, I mean, April is still pretty recent, so I'm not surprised it's yet to fully sink in, but it was really, really interesting to hear about all the techniques today and run through the framework that you're outlining. And we'll be recording, I've recorded the webinar, so we'll pass it on to all the rest of our members who weren't able to join. So unless anybody has any further questions, I think we can probably end it there. If you have any, quickly type them in now. If not, I'd like to give a big thank you to Leo, Trond, and Sarah and all the participants for the time today. It's been really interesting and a great chance to link up with Conservation International and their recent work on this. I've recorded the webinar, as I said, and I'll make it available on the IPECA Vimeo page if you'd like to listen again or share it with any of your colleagues. And please do let me know if you'd like the slides. And if you have any follow-up questions, you can send them through to me and I can link you up with Leo and Trond as appropriate. All right, thanks very much, everyone. Great to hear from you. Bye.
Thank you.
Thank you, Anna. Bye-bye.
Yes, thanks all.
Thanks very much. Bye-bye.